Well, we've come a long way in this biology course so far. So since we're at a good halfway point, it's fun to think about how do we know what we know? First of all, let me say that what we've learned so far in biology is just a tiny slice of what humans know about biology. And what humans know about biology is probably only a tiny slice of what more there is to learn. So we don't know that much about biology yet, but still we can take time to think about how do we know even the little that we do know. There are several reasons that we know what we know about the insides of cells. We know it mainly from a process called reductionism. Reductionism is the idea that if you can eliminate a lot of variables, you can figure out what is actually causing what. And it's very tricky to otherwise do that in biology because so many things are interacting. So, for example, when I study the protein kinesin, I can study its walking motion by looking at a neuron or a nerve cell under a microscope, which already is better than trying to look inside of an organism at the same kinesin because we've taken the neuron out and we can look at it more carefully. But we can also take the kinesin out of the cell and put it onto a glass slide and just look at the kinesin with adding in a little ATP to get it going. So that's a way of reducing the number of variables. Reductionism is very powerful. Sometimes it leads to the wrong conclusions, like in my own research. When you take kinesin out of its natural habitat, it slows down. And nobody really knows why it's a lot slower. And sometimes people just ignore that problem and just study whatever it is that they see on the glass slide and don't worry about it too much. But other things like that are reasons why we know what we know in biology. Facts and observations are not always what we're basing our knowledge on. We also have to make frameworks around those facts and observations, and that takes a lot of assumptions. Could we be wrong about those assumptions? Definitely. Do diseases have different causes than what we think? Absolutely. Proven over and over again that people think one thing causes a disease and then it turns out to be another thing. Ulcers in your stomach. People didn't know that they were caused often by little bacteria, and they missed that. Diseases like rabies and rickets and things like that, people didn't know the proper causes of those. So we're always making new discoveries, and we could be completely wrong. But a good exercise for you to think about this is what are you learning about, and what do you like learning about the most? What parts of biology or other things that you're learning about in your life are you really enjoying learning about? And why is it that you enjoy learning about those things? Is it because you also want to change what you are learning about and in life around you? For example, it might be interesting to learn about a particular disease condition because you want to learn how to cure that disease condition. So another exercise you could do is after you think about what you like to learn about, what do you want to change in the world around you? And think about how humans are changing life on Earth right now. Think about the good ways that humans are changing it, and think about the bad ways that we might be changing it. So learning about biology is critical because we want to learn how to change the world around us and how not to change the world around us, because sometimes it can be bad. One way that humans have learned recently how to change is a little story about an enzyme and a group of enzymes and a whole system of things inside of small bacterial cells that can change and edit the way humans have their DNA. So this is called the CRISPR story, and only in recent times have we been able to edit the catalog of choices that the DNA has. So we've known for a long time that many diseases have a genetic component to them, and we've been trying for many years to cure those genetic diseases by inserting DNA or changing the DNA. But it's been very difficult, and DNA therapy has been filled with many disappointments. So this recent technology that I was talking about called CRISPR is finally a way that we can specifically get in and edit the DNA in very discrete manner. And this is going to open up a whole new realm of therapies and just ability to change organisms so that we can study them more effectively. When you edit the catalog of DNA, you're editing the choices of what you can order. And so you can knock out choices, you can insert things and change choices and see how it affects the organism. If you did it in a human as they were living, you could potentially knock out and change a genetic defect and allow them to live more of a normal life. So as you think about that story, think about the dangers that might be caused by people going in and editing genomes and making designer humans, but also think about the powerful tool it might be for good. And that will give you an idea of what you have to learn in biology. If you're going a little deeper, there are a couple of leafs that might be interesting to you. For example, the leaf, why are microscopes so important for studying biology? Much of what we know and what I've taught you comes from careful observation under powerful microscopes. And then another branch that might be useful at this point to read is called What General Knowledge and Skills Are Important for Biologists to Have. Getting that list and knowing what that is will give you an idea of how you can be a well-rounded biologist.